Welcome, everyone. I'm Chu Fan Li, co-director of the Imperial Physics of Life Network. It's my great pleasure and an honor to introduce our seminar speaker today, Professor Nigel Goldenfeld from the University of Illinois, Urbana-Champaign. One of the best things about being a seminar organizer is, together with Tom Udrich, Robert Andres, and Chris Dunsby, is that I get to invite my favorite scientists to talk about their amazing research. And it's a bit like inviting your favorite musicians to play at a gig in your local venue. And today I feel very much like having an international rock star performing on this occasion. Nigel is a well-renowned physicist, a fellow of the American Academy of Arts and Sciences, and a member of the US National Academy of Sciences. He is also the recipient of the 2020 Neil Kandanoff Prize for his profound contributions to the fields of dynamical pattern formation, superconductivity, and fluid turbulence, together with creative developments and exposition of the theory of the renormalization group. So it doesn't mention that he is also a renowned biophysicist, and today he will talk to us about, is there universality in biology? So before I pass over to Nigel, Nigel has indicated that he wouldn't mind answering questions during the talk. So I would encourage you to post your questions on the chat, and Tom and Robert and I will monitor the chat and post questions whenever the opportunity arises. So, and also the talk is being recorded again, and if you don't want your names to be shown along with your questions, just post your questions anonymously. So Nigel, thank you again for speaking to us today, and I'll pass over to you now.
Well, thank you very much, Chu Fan, for inviting me. It's a great pleasure to talk at Imperial College, and nice not to have to fly in an airplane to get to you, although I hope one day I can actually visit in person. So I don't know the audience very well, but I understand you're a mixture of physicists and biologists and people who are somewhere in between those two end members. So what Chu Fan asked me to talk about was one of the more provocative talks that I have on my list of talks, and that is the question of, is there universality in biology? And so I'm going to go through fairly quickly a lot of separate problems. I'll try to delve into them in as much depth as I can to give you a flavor of what's going on there and what I think is interesting. And I'll also try to talk about some new work, but the goal is really to try to be provocative, make a discussion about these issues, and I look forward also to your questions. So without further ado, let me set up the laser pointer. So there we go. We have a laser pointer, and we're off. So here's what I propose to discuss. I want to answer these questions. First of all, do we learn new physics when we study biological physics? So everybody who is a biological physicist has probably run into this question at faculty meetings and things like this. You know, is biological physics really physics? Or is it just insanely complicated soft material science? What's new about doing biological physics? And everybody has their own different answer to that. Some people say, well, I'm going to make a better tool, a better microscope, or a better computer simulation, or something like that. But I think there's more that we can do, and I'm going to try to explain that. Then there's the question of, well, what are the universal phenomena in biology? And roughly speaking, I would say that there's two perspectives on this. One is that a biologist will say, well, everything is full of exceptions. Biology is full of exceptions. There's nothing universal in biology. Go away, physicists. And then the physicist might say, no, there's always a universal phenomena in everything. And it's useful to understand what those things are, because if you know what follows from rather general principles, then there's no need to try to explain that phenomena using some very idiosyncratic microscopic physics. And so we'll talk a bit about that. We'll talk about, do these universal phenomena reveal anything important, and concomitantly whether they hide important things. And then finally, I want to touch on, at the end, what do we miss by not understanding universal phenomena? And I'm going to draw on my experience as a condensed matter physicist which I know some of you are as well, and try to explain what there's perhaps a level of understanding of biological phenomena that is not commonly appreciated. So let's start with Carl Rose. So I'm sitting in my office at the University of Illinois at Urbana-Champaign. My office is not in the physics department, or I have one there, of course, but I spend most of my time at the Institute for Genomic Biology, or more precisely, the Carl R. Rose Institute for Genomic Biology. So Carl Rose was my colleague and collaborator for over 10 years. He was my mentor in working in the field of evolutionary biology and microbiology. And here he is, shown in characteristic fashion in his office on the front page of the New York Times, November the 3rd, 1977. So what had he done to get there? Well, as the caption says, scientists discover a form of life that predates higher organisms. Well, the predating higher organisms is not quite correct, but what had happened at that time, that was the date of publication of a paper in the Proceedings of the National Academy of Sciences, which reported the discovery of the tripartite structure of biology, namely that there is a third group of life, a domain of life as it came to be called, known as the archaea. And so here is the tree of life shown in this 1997 review by Norman Pace. And what you see here are the eukaryotes. So there's you. Virtually everything else on this graph is a microscopic, is a micro, but there's you and some charismatic megafauna. Here are the bacteria, and then here are the archaea. That was the domain of life that Carl Rose discovered, which is a third domain of life, and it is as different from the bacteria as you are. Completely different evolutionary history, and that was the big deal. Now, the question that Carl Rose was interested in was this question of the last universal common ancestor. In other words, at the root of this tree is an organism, or perhaps a community of organisms, from which all life that we can currently detect through DNA, through genomic sampling, is descended. And of course, you'll notice that the word last universal common ancestor, the word universal is going to appear a lot in this talk, even though it is ostensibly a biology talk. So what I showed you before was an example of a phylogenetic tree. And so here it is again, slightly in a slightly different representation, perhaps closer to the one that Carl Rose originally discovered. And there's many different ways to portray phylogenetic trees. So let me briefly introduce them. And what I propose to talk about for the next few minutes is the topological structure of phylogenetic trees. So when you do phylogenetic trees, you construct them by comparing genome sequences. And in fact, what Rose did was he was the first to look at molecular sequences, ultimately the 16S ribosomal RNA gene in bacteria and archaea, and eukaryotes, or the 18S gene. And from those gene sequences, for a hyperconserved gene, part of the ribosome that is present in every cell and part of the machinery of every cell, one can, by aligning the genome sequences and working backwards by doing comparative phylogeny, one can order them and create a sort of maximum likelihood picture of their relatedness, and thus from the leaf nodes at the edge, deduce what must have happened internally in order to deduce the evolutionary history. So the phylogenetic trees are indeed the trace of the evolutionary process, and they're very complex. So here's an example, a very clearly very complex, non-trivial structure of a phylogenetic tree. This is not a real one. This was generated by a simulation. And the way I want to think about this as a physicist is that the phylogenetic trees are the Feynman diagrams of evolution. And this is not just a fanciful construction, but as I'm going to show you, is actually mathematically what they really are. So let's talk about those trees. And what I want to talk about is what we can learn about the large-scale structure of the evolutionary process from the world's lines of all the world's species. So the first thing I want to tell you is that these phylogenetic trees are self-similar. And you may have noticed that just by looking at the chart, but we're going to look at it in a more quantitative way by rather than just getting an impression of some kind of fractality. So the first thing we're going to talk about is the topology. In other words, the structure of the edges and nodes. And by topology, I mean what is left invariant if we change the length of these edges here. And then the other thing I'm not going to spend a lot of time on, but we'll talk about the edge lengths of these lines between the nodes. Those represent, of course, the timescales in the evolutionary process. So how do we describe tree topology? So the way we're going to do it is the following. We're going to build a measure that tells me, roughly speaking, how many subtrees are rooted from a given tree. So let's suppose you start from this node over here, and you can see that there's one descendant from that going down over here. This node over here has these two nodes descending from it, plus itself, so that gets the number 3, as does this one over here. And then this one here has itself plus the 2 subnodes, and so that number is 7. So that's the node number for this particular node. But then we can also work out the cumulative size of the nodes descending from this. So basically what we do now is we take this one, and it has one node underneath it. This one here has itself, it has 3 plus these 2 numbers, so that gives you 5. And this one has these 2 numbers, 5 and 5 is 10, plus its own number, and that gives you 17. And so there's a recursive algorithm for computing the cumulative size of the subnodes from a given node. I apologize for those little beeps as messages come in. So that quantity we'll call C of A. And why is this quantity important? Well, let's take a look at what kind of trees one might expect by some kind of branching process that can create phylogenetic trees. So here is an example of a completely unbalanced tree. So each node goes down, and then the left node just goes down to the present day, and the right-hand node splits. It splits into 2. The left node goes down to the present day, the right node goes down, and then at some point in time splits. And these splitting events are speciations, because each node here represents a species. Here is a balanced tree, so each node just splits into 2, and this is a realistic tree. Now, what you can show, and I'm not going to show you the mathematics of this, is that if I compute how does the cumulative sum depend on the node number A, it scales basically as A squared. If, on the other hand, I have a completely balanced tree, it scales as A log A. And it's not difficult to show these things. And now you might ask, well, what do real phylogenetic trees do? So you can go and take all the phylogenetic trees in the world and process them. And what you find, and this is work that was first done by this group and rediscovered and done in a slightly different way by myself and Patricia Geraldo about 10 years or so ago, what you find is a power law scaling with an exponent about 1.4. So that's interesting. Real phylogenetic trees are somewhere in between these limits. And if you're interested, you can ask me in questions, but I'm not going to go into it now. There's a very interesting historical story about this discovery, which is in my paper in RNA Biology 2014. Okay, so the edge lengths, they also have a scaling law, and this one's a bit more complicated. So I'm not going to go through it in gory detail, but let me just give you a flavor for it. So what we define is a quantity. It's rather hard to get your mind around it. For every node, we ask how many tips follow from that node, and then we call that number I of k, and then we look at the edge length between I, the node I, and its particular branching point. So in that case, this one here, and then we sum up those numbers. So it's a bit of a mouthful. Let me just quickly show you how it works. If you don't get it the first time, it doesn't matter. It's not going to be crucial for the rest of the talk, but I just want to show you that there is a scaling law associated to this, which was discovered by my colleague James O'Dwyer, another British theoretical physicist who saw the light and is now one of the leading ecologists in his age group, and is also my colleague here at the University of Illinois. So what you do is you look for k equals 1, so that's all of these nodes here, and you can count up these numbers. So this number it comes up to 25. If you look at the nodes that have got 2 tips, so these ones over here, and count those up, you get 3, and then you can do the same thing for all the others. And then you sum up those edge lengths, and what you find, and you can show this, is that if you tried to just generate these branching patterns by some kind of Yule process, a kind of branching tree, a neutral model, you'll have an exponentially growing community, and this number will scale as k to the minus 2, the clade size. If, on the other hand, you have some kind of coalescent model, a Kingman coalescent or something like this, which says that these trees have common ancestors, then you will get a scaling law that goes as k to the minus 1. And when you look at real phylogenetic trees drawn from microbial communities, what you find is that they have a power law scaling with an exponent which is not very well delineated, but somewhere in this range over here. So the question is then, this topological measure and this metric measure capture the large-scale structure of evolution. And now the question is, where do these scaling laws come from? So we want to know where these scaling laws come from, and then of course what we want to know is, what do they tell us, if anything, about living systems? So before we talk about the universality of these laws and where they come from, let me remind you where universality comes from in physics, if there is any universality. So the most famous example of universality is what happens in condensed matter physics and in quantum field theory at critical points. So let me just briefly remind you what we're talking about here. We might have a magnet, typically an Ising model, where the spins can either point up or point down, and we can measure the number of spins pointing up minus the number pointing down, and that gives us the magnetization. And what we know in a magnet is, if you cool it down at 0 external magnetic field, at high temperatures, it is not magnetic, but as you go to low temperatures, it develops a spontaneous magnetization. It becomes a magnet, and it does that below some critical temperature Tc. And in fact, if you measure the magnetization as a function of temperature below that, it follows this curve shown in pink here, and this curve is not some kind of parabola or something, as you might have expected, but in fact is some rather singular function growing as T minus Tc to the power beta, where beta is some strange transcendental number below Tc. So that's one stylized fact. The other stylized fact is this. Let's suppose I have a magnet, and now I apply an external magnetic field. You might say, I've got a magnet, now I apply a magnetic field that will bias the spins in the direction of the magnetic field. How much magnetization will I develop by applying an external magnetic field? And if you asked a physicist on the street, what she would say is, well, you apply an external magnetic field, the magnetization, the response, is going to be proportional to the magnetic field that you apply. And that's indeed Curie's law. But that law, along with many others, breaks down at a critical point. And in fact, what you find is that if I sit exactly at the critical temperature and apply an external magnetic field, the magnetization that is induced is not proportional to h, but scales as h to some power, which is conventionally written as 1 over little delta. And what was realized in the 1960s by Ben Widom and Leo Kadanoff, and was in fact the foundation for the renormalization group, was that these laws followed from a similarity formula that describes the thermodynamics of the system. Namely, the magnetization, ostensibly a function of the temperature and external magnetic field H, was in truth a function of the ratio of them with some particular scaling laws as shown here, some beta and a new exponent, capital delta, and some universal scaling function. Now, I'm not going to go into why this is so or how the renormalization group tells you where that comes from. I'm going to skip over this slide, but I want to just show you that it works. So here is the universality. These are experimental data with a magnetization plotted in the way that theory predicts. the temperature scaled in a way that theory predicts. And these are data from 5 different magnetic materials, all plotted in this way. And you see that those data all overlap and fall on a one-parameter universal curve. And if you can see it, this solid line through here is the renormalization group prediction with no adjustable parameters. And so this is the famed universality at a critical point. It's very, very remarkable and striking, and it revealed very deep physics. Now, you might say, well, that's great. So what I've shown you then is that, say, the Ising model or Heisenberg model of the ferromagnet gives a very precise prediction, which is in agreement with experiment and with no adjustable parameters, and that's remarkable. But it's really a little bit more surprising than that, because the model is not the model of spins, an Ising model or Heisenberg model of spins pointing up and down. It's really a model of a model of a model of a model of a model. And why do I say that? Well, at the lowest level, you have, say, quantum chemistry and the atomic orbitals and so on. Then you have the electronic structure. Then you have the description of the interacting dipole moment, which is, say, a quantum Heisenberg model. Then you say, well, the quantum model is too hard to solve, so you make it classical. Then you develop the Landau theory for that. And then finally, you do a normalization group and you get a precise prediction in agreement with experiment. So you've got the sequence of models, and you might say, okay, it works, big deal. Quantum electrodynamics also works to 10 decimal places accuracy. But the thing that is really remarkable about this is that every step here, every modeling step, is not a systematic approximation. It's not one that you can justify by small parameters and the usual type of things that physicists are used to. These are all non-systematic approximations, and yet these brutal and crude approximations that you make, say going from quantum to classical, and from here ignoring the electronic band structure and things like that, none of those things matter. And that's why this was such a shock, and why, when this was understood and solved, Ken Wilson won the Nobel Prize. So where do these strange power laws come from? So let me explain that to you, and we're going to return to our biological example in just a minute. So where it comes from in condensed matter physics is the following. When you observe anything in condensed matter physics, you're doing a scattering experiment. And what scattering experiments measure is a structure function, or if you like, a correlation function. So here's the correlation function as a function of a wave number at the critical temperature. And what you can show is that it has the following form. If you look, in page 194 of any good book on normalization group and statistical mechanics, you'll find this formula here, and it says that the dependence of the correlation function in wave number goes as k to the minus 2 plus some weird transcendental number, which is called an anomalous exponent. And it's a surprising formula, because the correlation function has units of length squared. It's easy to show that. And so what you would expect, you'd say wave number has units of inverse length, so you would think that it would just go as k to the minus 2. But it goes as k to the minus 2 plus this anomalous exponent eta. So what's happening here is you have scale interference. Even though the correlation length in the system in its critical point is very large, the system still sees the existence of the lattice spacing on the scale of angstroms, and that length scale enters in a singular way as shown here. And because it enters in precisely this way, it cancels off the anomalous behavior from this strange anomalous exponent, and it restores dimensional analysis.
So Nigel, there's a question from Tano Norand. What is a systematic or non-systematic approximation? So I guess that refers to your previous slide.
Yes. So what I mean by a systematic approximation is one where I can give you a bound on the error that I'm making with an approximation. For example, I might be doing a perturbation series or some sort of expansion. And so I know, here's the first order term, here's what the second order term is, and I know exactly systematically what those other terms are. So, and usually if I'm doing, say, perturbation theory, like we know in quantum mechanics or classical mechanics, I can say, if you want to do a bit more work, you can get the next term in this series, or the next term, or the next term, or the next term. That's not the case in these type of things. Okay, so hopefully that answers the question. So the point about this is that what you would have guessed is that that on large length scales, the lattice spacing doesn't matter. You're looking at a system on the scale of the Earth's diameter, and why would you care about something that happens on the scale of angstroms? So you would think then that the exponent, the scattering function, will scale as k to the -2, but in fact, it's not true. So what it means is that you cannot ignore the lattice spacing. You cannot just say, because the length scale I'm interested in is much larger than the lattice spacing, I can just set the lattice spacing to 0, which is what physicists normally do when they sloppily do dimensional analysis. So the point is that the system remembers the small scale details, even if the correlation length of the critical point is diverging to infinity. So the question then is, this phenomenon is called scale interference. And the question then is there scale interference in evolution? So let's think how that could happen. So here's our caricature of evolution. So here's our phylogenetic tree. And these nodes represent speciation events, and that those speciation events happen due to ecosystem dynamics. There's some mutation perhaps, or some other genomic event. The environment selects some new variant, and that new variant eventually forms a new species, and so you get this speciation event as shown here. And so there's two timescales here. One is ecological, which is on a short time scale, and one is on evolution, which is on the long time scale. And so when we draw this phylogenetic tree, we're implicitly ignoring ecology. So we're setting the ecological time scale over the evolutionary time scale to 0. And that's, I'm going to suggest, is like setting the lattice spacing to the correlation length equal to 0 in a critical point. Now, we know that that's an oversimplification, because we know very well that there is an intimate connection between evolutionary and environmental timescales, and there's a whole field of ecology known by the buzzword rapid evolution, where what one studies is how the environment selects on the population structure, and the population then adapts to the environment, creates new metabolic or biochemical niches, and those themselves then alter the environment. And so there's this feedback between these things. And in other research in my group, we've studied how this can affect, can be seen in ecological dynamics. For example, in anomalous phase differences between predator and prey in an evolving ecosystem. We're not going to go into that, but the point is that evolutionary events can change the entire trajectory of an ecosystem. And so we might ask that because there is this feedback between ecology and evolution, perhaps the timescale separation that we're assuming isn't valid. So that's one point. The other point I want to make is that we can think of ecological interactions as particles and fields. And so the next couple of comments are really intended for those of you who come from theoretical physics in the audience. If you don't fully understand the nuances of what I'm saying, don't worry, there'll be plenty more things to follow. It's not essential, but I want to make this point. So let's
Nigel, to interrupt.
Yes.
I guess there's also the issue with the environment, that it also happens at many different time scales, right? I mean, this is sort of the whole debate about weather versus climate.
That's
you
correct.
can think of it almost at a daily scale or a weekly scale, and then things happen on a centuries or even 10,000s of years, depending how the sun activities and so on. I guess it makes it as well very hard with the environment.
That's right. But we can talk about long-term environmental changes as being the ones that are more likely to generate speciation events, and short-term ones which are more likely to be small modifications of the environment that may change population structure, but may not necessarily lead to evolutionary events.
Mm-hmm.
So I talked a minute ago about predator-prey dynamics. So predator-prey dynamics is something that we're super interested in my group. And the idea here is you have a predator, which is A, B is the prey, E can be the food or some sort of empty state. And predation is really what happens when a predator meets a prey, and then with probability P eats the prey and produces two predators. And so these reactions here describe the reactions that describe a predator-prey ecosystem at the individual level. And we can also write these things down we can formulate these things in classical statistical physics using what's known as the Doi formalism and map the stochastic modeling of these predator-prey ecosystems from a master equation point of view into a quantum field theory through annihilation and creation operators. And when one does that, one can write down Feynman diagrams for that. So the solid lines represent the predator, the wiggly lines represent the prey, and each of these interactions here has a Feynman diagram associated to it. For example, this one here is that a prey and a predator meet, that something happens, and at the end out comes two predators, and the prey is being eaten. So this is the Feynman diagram that corresponds exactly to this process. So this is a sort of particle wave duality. And you can make these things very, very precise. So in quantum field theory, when we talk about Feynman diagrams, we talk about vertex renormalizations, such as in QCD. And when one does population dynamics in this way, as I do in my group, we look at spatial patterns and so on using these kinds of methods, one sees again these sort of vertex renormalizations that tell you about microscopic ecological processes. Now, if this is new to you, what I've just told you, it's not very widely known, and I would recommend it, not necessarily my own papers, which are not terribly pedagogical, but some wonderful papers by Uwe Teuber, a colleague of mine at Virginia Tech, who's written many pedagogical articles, a wonderful book on critical dynamics, which talks about all of this and how one can apply field theoretic methods to studying spatially extended ecosystems and evolutionary systems with demographic stochasticity. So I just want you to know that this idea that these phylogenetic trees represent some detailed ecological and evolutionary structure is not fanciful, but can be translated into mathematics. And in my own group, we've used these things to study evolutionary ecology. Now, but going back to the biology, so where this comes in is in what's known as niche construction. Typically, when we think about evolutionary ecosystems, what we think about this, O represents the organisms and E represents the environment. So typically we think of that the organisms have some equation of motion which depends on the organisms and depends on their environment. And we typically think of the environment as just changing under its own dynamics. And what people realized about 25 years ago or so, maybe a little bit more than that, was that, of course, organisms change the environment, as I hinted at before, and so there should then be a dependence on the organisms in the equation for the environmental evolution as well. The environment selects for the fittest phenotype, roughly speaking. Organisms change the environment, and so evolution becomes a collective behavior. And there's wonderful examples of that, and none better than the fact that you're alive today. And you're alive today because you can breathe oxygen, and oxygen was something that was a poison more than 2.6 billion years ago. It was produced by cyanobacteria that produced this as a metabolic waste product. The oxygen accumulated from this great oxygenation event and led to the current high energy biosphere, which enables anaerobic organisms to die and aerobic organisms to evolve. And because oxygen is basically rocket fuel, it enables high energy organisms such as multicellular eukaryotes to evolve, where previously there was really just microbes and so on. So this is an example of organisms changing the environment, in fact, on a global scale. So now I want to show you very quickly a very caricature model of niche construction. A caricature in the same sense that the Ising model of a magnet is a caricature of what happens in a real magnet. So we're going to talk about the niche as the position of a species in its ecosystem. And I'll show you what I mean by that in the model in the next slide. And niche construction says that there's some kind of mutual interaction between the species and its ecosystem. and the survival and the diversification of a species depends on its niche, in other words, the ecology. And the niche of a species is basically correlated with its ancestors, and so that tells you that there's a connection from generation to generation, which has some evolutionary aspect. So this minimal model, and again, I'm going to go through this quickly because I just want to show you the results. I don't want to go into details about them because I've got a lot more other examples I want to show you. But basically the way it works like this, we construct a phylogenetic tree with 3 numbers associated to each node: the available niche, the speciation rate, and the extinction probability. And the idea is that the niche is the total available growth space that an organism has, or if you like, the evolutionary degrees of freedom of the organism. And so the physics of this is that if you have a large extinction event, there are now many niches available that can be colonized by the successors. And so as is well known, there are bursts of diversification that happen after extinction events. And so this model tries to capture that. So an organism with a large niche value, this number n, has then a large number of possible ways to adapt to the environment, and that then translates into something that relates to the, I'm just turning down my phone here, that relates to a model of the evolutionary dynamics. So what we say then is, how does the available niche number evolve? And we assume that it changes to a geometrical Brownian motion with some fluctuation given in the way shown here, and that captures the sort of inheritance aspect. We're going to look at the speciation rate as being a function of the niche value. So the more niches you have, the more likely speciation is to occur, and then we have to bound the rate of growth of the tree, so there have to be extinction events as well, so that you don't get an explosion, an unbounded explosion of the number of species. So this is a kind of dynamical system that describes a niche construction model for eco-evolutionary dynamics. And what one finds is when one looks at the ratio, the C as a function of A, one sees that as one varies the fluctuations in the system, one sees plotting C as a function of A, one sees the dynamics approach this straight line here with a scaling law of about 1.5. So this model captures the scaling law that I showed you before. And similarly, if we look at the cumulative edge length distribution, again, I'm not going to go through this in detail. This is the same quantity that we talked about earlier to do with the microbial communities. One again sees a scaling law as a function of the subtrees. These events here, this is due to undersampling of large subtrees. I don't want to go into this, but these scaling laws are robust. So what I've shown you then is that the real data are recapitulated by these very simple coarse-grained models that capture the topology and the edge length distribution by this niche construction process. So are these really anomalous parallel scalings in the sense of critical phenomena? I'm going to skip through this in detail. What I want to show you is this graph here, which is the analog of the data collapse graph that I showed you earlier for the magnetization.
Nigel, actually, sorry to interrupt again. I wanted to ask about you showed the scaling law as well of the data. I think the power was 1.44, and you had this experimental result. I think it was 1.51. Of course, the experimental result also, the experimental result is just it's just for phylogenetic trees. It's just a maximum likelihood estimate. Is it right? And I guess
that's correct. I mean, the group that first published these results gave the exponents 3 significant figures. I would give it to less significant figures and with a larger error bar.
Exactly. I was wondering about the error bars. I guess one could think of it more like a Bayesian posterior, and then one could estimate, I guess.
Yes, I think that's right. I wouldn't draw the distinction between 1.4 and 1.5. I don't think is a relevant one. And as shown here, the correct way to plot the data is to do the data collapse scaling and look at the universal scaling functions rather than power laws. Because as we know, even in critical phenomena, power laws are very difficult to extract but universal scaling functions are much better because you're looking at a whole function rather than a single number. So I agree with your point. So the main point is not that we're trying to get exactly the number, but we're just trying to show that there is a robust scaling here. Just like in the same way you can see that with a magnet. Now, in a real magnet might not be an Ising magnet, or it might not be a Heisenberg magnet. It might have some anisotropic interactions or some long-range interactions that will slightly change the universality class. So the point is not, can you reproduce the exact exponent, but can you recapitulate the actual phenomenology? And as I've shown you, one can.
So sorry, Tom Aldrich has a question.
Yes, go ahead.
If niche evolution is random, is there any feedback from the organisms?
There is randomness in these models, but there's also feedback from the organism. So it's a mixture of the two things. It's a stochastic dynamical system, but yeah, there is feedback.
Okay, so you talked about the niches being it looked like there was no information coming in from the system into how the niches change, but actually you're saying there is information coming from the system as to how the niches change.
Yes, there is, because the speciation rate depends on the niches, and then the niche dynamics has some stochasticity in it, plus some inheritance.
Oh, niche dynamics, okay, fine, okay, thank you.
Thanks for the question.
So that's another question from Ricard Alert. Okay. Are the experimental scaling laws reproduced only in the limit of large fluctuations? If so, why?
That's not quite true. The reason that you get these scaling laws in statistical physics, they're associated to large fluctuations, but they're probably more properly thought of in terms of scale interference. In other words, even systems that have no fluctuations at all, deterministic systems, have these anomalous scaling laws. And there's work that I did on that 25, 30 years ago. And if you look in chapter 10 of my book, that is explained in detail. So it's commonly thought that the scaling laws are associated with large fluctuations, but they're not. They're associated with singular asymptotic limits. Now, in this particular case, there are large fluctuations because you have these geometrical Brownian motion processes which come from the evolutionary dynamics. So hopefully that answers the question. So let me move on. So what I've shown you is that the scaling laws can be described. I haven't proven to you that this is the only way you can get them, but this is the only way that I've been able to think of, which is they come from this interplay between ecology and evolution. And they tell us that evolution is more than just mutations. One must take into account ecological dynamics on scales that are shorter than evolutionary ones, and that relate to things like competition, predation, range expansion, metabolic cross-feeding, and other ecological processes. Okay, so now I want to switch gears and talk about universal biology. So here is a headline that came up on the news, maybe how many years ago? 6 or 7 years ago. Cassini spies the wind-rippled waves on Titan. Titan has lakes, and they're not water, but Cassini was a wonderful satellite that made many incredible discoveries. And this was important because previous year, the headline was Mystery of the Missing Waves on Titan. In other words, why aren't there water waves on the oceans of Titan? And the answer was, well, there were, we just didn't detect them. Now, the fact that there were waves on the ocean of Titan, and we knew that they were missing, is because we have a general theory of two-phase fluid flow interfaces that predicts what those waves should be, what the dispersion characteristic should be, what their amplitude should be, and so on and so forth. Here's a headline that I never saw. Mystery of the missing life on Titan. And finally, Cassini spies long-sought life on Titan. Now, we didn't see that, but I'd like to have done. In fact, Cassini may have seen evidence of life on Enceladus, a different outer solar system moon, but that's a whole other story. And why is one of those headlines a serious scientific one and one a kind of joke? Well, the point is that we don't know if there's missing life. because unlike the situation with water waves, we don't have a theory that predicts the existence of life as a physical phenomenon, and nor do we understand what sort of life can arise in different environments, generally speaking. So
hey, Nigel, there's a question I think about the previous part still of your talk. Maybe I can ask this quickly, as it was by Jeff Fredberg. What about real physical trees, such as botanic trees, blood vessel trees, airway trees? I guess it's about the scaling.
Those also do have scaling laws associated to them. I haven't worked on it myself, but other people like Eleni Catafori and others I'm blanking right now have done work on that as well. That's a very good question. There are scaling laws, and they can even be understood in terms of the growth processes. There's ones in leaf veins and things like this. So yes, there are scaling laws associated to those things too, and to river systems and all sorts of other trees. You're exactly right. Okay, so for this part of the talk, universal biology, we're interested in general principles of life that don't depend on specific carbon chemistry details. And so one would like a theory of universal biology divorced from idiosyncratic details, and this will involve abstractions. And you might say, well, how can abstractions be useful in real biology? And the answer is that they can be. So let me talk about universal biology. And pretty much what's shown on the slide is what's known about universal biology, i.e. not much. So let's talk about something which is known about, and that is universal computation. So here's a computer. This is roughly what I'm projecting my talk on at the moment. Here's another computer, and you guys can go and see it, because actually it's pretty much round the corner from where you're sitting if you're sitting at work in the Science Museum. This is Babbage's computer, which actually can be made to work, and there's one at the Museum of Computation in Mountain View in the US. And the point is that these two things are the same. This thing is made out of glass, plastic, and silicon. This thing is made out of cogwheels, springs, and levers. But the point is that they're actually the same device. They are instantiations of a Turing machine with a von Neumann architecture. One is built with technology of the 20th century, or maybe the 21st century. One is built with the technology of the 19th century, but they do the same thing. So the medium is not the message. And here's another example. These are the first computers that John von Neumann built at the Institute for Advanced Study in the 1950s, built out of thermionic valves and relays and switches and things like that. So this is very slow. This is pretty slow. And this thing is frustratingly slow at times, but occasionally works. So the question then is, are there new physical laws that describe biological systems which are abstracted rather than ones that are based on, where we try to understand them in a level of too much realism? And Schrödinger and Delbruck were some of the earliest physicists to work on biology, and their motivation was to find new physical laws. Now, where would such laws lie? Well, they thought the answers would lie in quantum mechanics, and maybe they're right about that, but the answer that I would give is that these new laws are the emergence and existence of life itself. So here's a real question. How is it that matter can self-organize hierarchically to create replicating evolvable structures? How do molecules come to life? So we're used to, in statistical physics and soft condensed matter physics, the idea that we can take a bunch of molecules and they can self-organize into colloids or block copolymer patterns or anything like this. But how does matter self-organize and actually create replicating and evolvable structures? And to understand biology in the same sense that we understand physical phenomena, we need to understand the existence and not just the specific realization of biological phenomena. Now, what I mean by that is the following. When we understand physical systems, we first of all understand how they can arise, the general principles, for example, that govern scaling laws. And then we understand the specific laws that apply to a particular system, say magnetic interactions, or in the example I showed you, niche construction. So what is the sort of broad class of dynamical systems that are living systems? And the answer I would suggest is the dynamics of systems that can reprogram themselves. And I'm not the first person to suggest this. The first person who probably thought about this was Niels Barichelli. So whilst John von Neumann's engineers were building the first computers in the Institute for Advanced Study in the 1950s, in their day life, they were designing computers to solve the equations of hydrodynamics to build hydrogen bombs. Niels Barichelli understood that you can make programs that act upon themselves. In other words, misusing the fact that data is a plural noun, program is the data is the program. These are programs that self-modify themselves, and he built the first self-modifying programs in what we today call machine code, and built and did the first experiments in what today we would call artificial life. And this is an image that I got from George Dyson. So this idea of self-programming systems, that living systems have the capacity to evolve, evolving systems can reprogram themselves, and so they're able to respond to perturbations by creating new functionality. And that's something that's very different from physical systems, which are very fixed or predetermined equations of motion. And perhaps this is a speculation, but I would even go so far as to say that the future state of an evolvable system might be inherently unpredictable, perhaps even Gödel undecidable. And the reason is because evolving systems can essentially change their dynamical equation of motion. Their phase space is not even defined at the outset, but is something that emerges from the time dynamics. And so because the phase space isn't known in advance, it's hard to say what kind of states they're going to visit in that phase space, and therefore the dynamics would be Gödel undecidable. This is a feature that makes it very difficult to control evolving systems, and we know that that's true because whenever we intervene in biological systems by trying to develop antibiotics, by trying to invent pest control, and so trying to do chemotherapy and so on, those interventions fail because the system evolves around our intervention. Now, in biology, we typically have the inverse problem. And the inverse problem could be stated in the following way. Suppose I were to give you a modern computer, say an iPhone or laptop or whatever, could I work backwards from that, from looking at that machine, and deduce the abstract theory of computation due to people like Turing and von Neumann? Obviously, it would be very hard to do, but that's what we're trying to do in biology as physicists at a high level. We have the inverse problem. The biology is already created. We already have very advanced biological systems, and we want to understand what is the abstract theory of which the chemistry is the instantiation. And if you could understand that abstract theory, that would underlie all systems that exhibit the characteristics of life. Now, I was thinking about these sorts of questions because for 6 or 7 years, we were funded through the NASA Astrobiology Institute, trying to understand what kind of life could exist in other planets. So let me show you some very quick examples about universality in biology. And I'm going to go through these very quickly so that I get to the end, which I want to get to, and still be respectful of your time. So here's an example of an abstraction. This is the Universal Genetic Code. It is an abstraction that encompasses many critical biochemical things that go on inside living cells. And it's remarkable that you can put down the whole process of a gene expression onto a simple table that can be used to understand how genes and proteins, how genes create proteins. This genetic code actually has its own dynamics. I'm not going to go through work that I actually did on this about 10 years or so ago with Carl Woese. The genetic code is not just some random code at random, just developed at random. It's not an accident, as Francis Crick called it, but actually it has very, very clear evidence of a strong optimization. I'm not going to go through this. I'll be happy to answer questions on it. We know that the genetic code evolved, in fact, through horizontal gene transfer, and so so again, this enables us to understand some of the features that we see in real biology at a fundamental level in terms of evolutionary processes. I'm going to skip this one. This is one that I want to just point out to you, which is the universal statistics of gene expression. So you can have genetically identical cells which exhibit different protein copy numbers. That's due to stochastic gene expression. And one would expect that protein expression would depend on genetics, the environment, epigenetic markers, metabolism, gene regulation would be very idiosyncratic. But when you look at the data, and these are data from Hannah Salmon's group from about 10 years or so ago, one looks at the probability distribution for protein copy number for a variety of different systems and different organisms. They all fall when scaled correctly onto one universal curve with an interesting scaling of the variance with respect to the mean. And I'm not going to go through this in detail, but if you're interested, I can tell you a little bit more about it. But what we learn from these data collapse laws that were very surprising when they were first discovered is that the protein production has a negative feedback control. And this is now a very active area in the cell biology, understanding how cell growth and cell division occurs in the cell cycle through interacting networks of reactions. Here's another one that's probably not known to most of you, which is universal metabolism. So this is a bioreactor fed with glucose and yielding oxygen and methane in a steady state. And so you make such a bioreactor, it produces methane, you measure the methane that comes out of it, and you look at this thing and you say, what are the microbes that are going on? What are the microbes that live inside this bioreactor? And so you go and sample that bioreactor, and you sample it once and you get a pie chart that shows you the community structure. And then maybe you're a little bit crazy, so you say, let's keep this thing running for a couple of years, and let's measure every few months the structure, the community structure, the microbial structure, the taxonomy of my bioreactor. And what you find is that there are massive fluctuations in the population structure. These different colors are different groups of organisms, microbes, doesn't matter what they are. What you can see happening is that as a function of time, the community structure is changing significantly, yet the metabolism is completely insensitive to these large taxonomic fluctuations. There's a functional redundancy, which is reflecting some kind of emergent metabolism. And this isn't just something that happens in the lab. Here's a paper from Luca and Michael Derbley a couple of years ago in Science, again, looking at functional redundancy in the global ocean microbiome. In other words, the taxonomic composition of the system is a very weak predictor of the ecosystem function. And that means that if we're going to understand the Earth's biosphere and its response to global climate change, we need to understand the emergent metabolism of microbial communities. So is universality useful if we find it? Well, let's talk about that. So here's an example, and the answer is yes, it is. So this is an example of universality. It's a scaling law in ecology, first discovered by bird watchers in the United Kingdom in the 1850s. And this is one very old plot showing you the number of plant species as a function of area. So you take a patch of an ecosystem, you measure how many plant species there are in it. Now you double the size of that. How many more species are you going to find in a somewhat bigger system? Well, you might have expected that the number of species would double if the area doubles, but that's not what happens. In fact, roughly speaking, the number of species scales like the area to the 1/4 power. And this is important because every day you might look in the newspaper and you might find that a whole bunch of forests have been burned down in the Amazon and a whole bunch of species became extinct that day. How do we know that? Well, we know that by using this kind of scaling law. So there's an example of how this scaling law is useful. Here's another example. And the answer to this question is the scaling law is not useful. So this is another scaling law in ecology. This is Barra, Colorado Island, where one measures the number of tree species in great detail and is a gold mine for ecologists. What one wants to know is what is the species abundance distribution, the fraction of species with a given number of individuals. So you plot that as a graph, and so this is the experimental data, and then you ask yourself the following question. Is this species abundance distribution that you observe, is it the outcome of some random process, just some species has to be the most abundant one. So whatever it is selected by chance, and then the next species has to be the next most abundant one. What would be the probability distribution based on randomness? That would be one. Way of thinking about this, a kind of neutral theory of ecology. The other theory that might be one which is more based on the ecological niches, trying to understand the microscopic processes that go on there. And when you take these two very different theories of species abundance distributions, you find that within experimental error, they can both account for the data. And so it doesn't help you understand what created the structure that you're seeing in this ecosystem. So the upside of universality is that you can make predictions based on a minimal model. That's great. But the downside of so an example of a minimal model is my model of the niche construction, or in physics, the Ising model of phase transitions. The downside of universality is that statistically similar results can be obtained from microscopic models that are very different in their assumptions. So when you have universality at high levels of description, that can preclude understanding the lower levels of description. If you see something scaling in the Ising law, in the Ising model universality class, that doesn't mean that it is an Ising magnet. In fact, it doesn't even have to be a magnet at all. The liquid gas transition has exactly the same scaling laws as the Ising model for magnetism, and the liquid gas transition is not even a magnet. Okay, so what do we miss by not understanding universal phenomena? And this is the last topic that I want to talk about. So I want to answer this question by showing you an example from condensed matter physics, and that is the history of superconductivity. So here are some of the milestones in our understanding of superconductivity. So superconductivity was discovered in 1911, the discovery of zero resistance. In 1933, excuse me, it was discovered that superconductors also expel magnetic fields. That's known as the Meissner-Ochsenfeld effect. That was 1933. In 1955, the vortex lattice structure of superconductors, type 2 superconductors, was discovered. And in 1962, the strange quantum interference effects known as Josephson effects were discovered. And these ones here were predicted theoretically and then very quickly discovered experimentally thereafter. Now, the question I want to ask is, why did it take so long to make these discoveries? And in particular, this one over here, from going from here to here was 22 years. Why did it take so long to discover this? Well, I teach condensed matter physics at my university. I can derive with a few lines of algebra that fits onto one page all of these effects. But it took the scientific establishment 50 years to go from the discovery to the elucidation of Josephson effects. And this is, and the reason is that we had the wrong level of description in physics. So let me talk about that. In superconductivity, one can have a description that depends on quantum chemistry and the material science. That's the lowest level of description. There's an intermediate level of description, which is the Bardeen-Cooper-Schrieffer theory for interacting Cooper pairs, which was formulated in my university, which is a very famous university in condensed matter physics, and this is one of the things that it is known for. And then at the higher level of description is a description based on symmetry, so-called Ginzburg-Landau theory for off-diagonal long-range order coupled to the electromagnetic gauge field. So these are different levels of description. This is a very abstract, high-level one. This is one that at least recognizes that electrons exist and form Cooper pairs, and this is one that is ultra-detailed. In biology, we can think of things in the same way. At the lowest level of description, we have atoms and molecules. At the highest level, at a higher level of description, we have biomolecules like DNA, RNA, proteins. We have the elasticity theory of DNA. We have the phase transitions for liquid-liquid intercellular complexes, and all of this kind of wonderful molecular biology and cell biology. And then at the highest level, we have the dynamics of evolving systems. These levels of description answer different questions. So in superconductivity, quantum chemistry and material science can explain for you how can you make a specific material that instantiates the BCS mechanism for forming superconductors. The BCS theory for interacting Cooper pairs answers for the question is, what is the basic mechanism by which superconductors function? In other words, in this case, how electrons form a bound state through the exchange of virtual phonons. And then the Ginzburg-Landau theory, the symmetry theory for off-diagonal long-range order and electromagnetism, that answers the question, why does the phenomenon of superconductivity even exist at all? And these different levels of description answer different types of questions. If we go back to biology, when we look at these different levels of description, what question does it answer? Atoms and molecules answer the question of how specific biopolymers interact, fold, undergo template-directed synthesis, and so on. This level of description answers the question of what are the basic functional cellular processes of life. And this level of understanding, if we had it, would tell us, why does the phenomenon of life even exist at all? Now, knowing these questions tells you about the level of universality. In superconductivity, quantum chemistry and material science tells you about specific materials property. The BCS theory for interacting Cooper pairs applies to what are called weak coupling superconductors. It doesn't apply to the high temperature superconductors, for example. And then the Ginzburg-Landau theory, the symmetry-based theory for off-diagonal long-range order and electromagnetism, that applies to all superconductors. So similarly, these levels of description would apply to biopolymers, the physics of subcellular components, and all of life, different levels of universality. So why do we need these different levels? Well, let's have a look at what happens if you don't have the Ginzburg-Landau theory for superconductivity. The BCS theory won the Nobel Prize for its contribution for understanding superconductivity, but it got things wrong. We didn't understand the Meissner effect, the vortex lattice, and the Josephson effect. Eventually they were understood, but the microscopic description failed to predict the response of superconductors to electromagnetic fields. Once you have this level of description, the one that I teach in my classes, you're able to explain all these things within a very few lines of algebra. Similarly, if we have this level of description in biology, if we don't have it, we won't be able to predict the response, not to electromagnetic fields, but to selective perturbations. And so what that means is, when we treat biological systems as physical systems, we intervene with them by prescribing antibiotics, insecticides, herbicides, chemotherapy. What we always encounter is the system evolves and it defeats our intervention. We have a dramatic failure to predict the response to selective perturbations because we don't really understand the top-level description of evolving systems. So by regarding superconductors as collections of atoms, we're missing the emergent laws that act at the system scale and govern large-scale response to electromagnetic fields. We know how to solve that problem, but we didn't for 50 years. It took physicists 50 years to discover that. By regarding biology as complicated physical systems, we're missing the emergent laws that act at the system scale and govern the large-scale response to control perturbations. We do not know how to solve this problem yet. This is a high-level problem, one that is ideally suited for physicists to contribute to. So some of these perspectives are described in these articles here, and which you can just download. And with that, I'm going to conclude. So here are the questions that I wanted to start off with. Do we learn new physics when we study biological physics? And the answer is yes, we do. We understand systems that have large fluctuations. They can self-organize into evolvable, modular, self-programming structures. By studying biological physics, we are expanding the scope of physics and dynamical systems. What are universal phenomena in biology, and are they important? Well, things like the genetic code, homochirality, which I didn't talk about, patterns of gene expression, the distribution of species, metabolism, and there's many more. Do they reveal things that are interesting? Yes, they do. The phase diagram of life, the cell structure and principles, but universality can obscure microscopic lower levels of description. And what do we miss by not understanding the universal phenomena? We miss something very important, something that's a big problem in today's world, and that is how biological systems respond to perturbations and how we can control them. We haven't learned to do that because we still think of biological systems as physical systems. And so that is a wonderful task for the future. So I'll leave you with this quote from Stan Ulam: "Ask not what physics can do for biology, ask what biology can do for physics." And my answer to that question is, by studying biology, we are expanding the scope of physics. So with that, thank you very much for your attention.
Thank you very much for the thought-provoking talk, and I'm sure this presentation will be gold mine for people to look for new ideas. And the so the talk is recorded, so it will be on our website. So I know Nigel already said that he is quite busy today. So do you have to go now, Nigel?
I can stay on for maybe 10 minutes, something like this.
Okay, fantastic. So
well, I'll be happy to take a couple more questions if there are any.
Okay, great. So we have a question from Tim Baraklar. Where does natural selection fit in?
So I'm not sure what you mean by natural selection. You mean the process of evolution? I mean, I think the thing I would say is this: it's people use the term natural selection. In fact, Alfred Russel Wallace kind of criticized Darwin's use of that term. Alfred Russel Wallace was the co-discoverer of this idea, and we think of selection because we think of Darwin was thinking of pigeon breeding and dog breeding and so on, where somebody is doing the selection. But my point is that really you can't think of it as here's the thing that's doing the selection, here's the thing that's being selected. You can do that in some idealized situations, but on the large scale, the biosphere is one connected dynamical system, which is doing its own selection as it goes along through the interaction between the different scales of it. So I think natural selection is certainly a way we think about a population genetics and so on and so forth. But in terms of understanding the high level question of why does life even start at all? How does evolution happen? That is natural selection is an outcome of that rather than the explanation for it. I don't know if I've answered the question, and I think we could talk for hours about this interplay, but I've tried to give you some flavor of what I think.
Well, I think there's an alternative way of thinking about that would be that natural selection is the definition of a living system, and I'm thinking of work by Eigen and that.
Excuse me, by work by whom?
By Eigen, Manfred Eigen.
Yes. That's right. I think the point is that I think what he intended, it was the idea that the evolutionary process is the definition of a living system, and that's one I very much would agree with. I think natural selection is a subset of the evolutionary process, but not the whole one. And of course, it depends how you're defining natural selection. If you're thinking of natural selection as this whole evolutionary dynamics between the ecosystem and the organisms, then yes, then I agree. My point is that people sometimes think of the definition of life as being systems that can replicate and so on, and I think no, it's the evolvability of living systems that is the defining characteristic. And I think that's what Eigen was talking about. And I know from some correspondence with him and reading some of his papers, in fact even ones where he actually quotes some of my own work, I think that's pretty much the picture he had in mind. Thank you.
Okay, so the next question by Roman. Can you provide, that's not a question. So Yin Tang has a question. Back to the first part, do you expect there is scaling laws for the evolution tree in faster timescale like influenza and COVID-19?
Oh, no. It'd be very hard to see them on that. And the reason is that the scaling laws require there to be very large phylogenetic trees. And so if you're looking at, say, influenza or COVID or something like this, the trees don't have enough depth that you can really see any really believable scaling law. You need to have several decades. So we had to do this over the whole evolutionary history of life rather than just looking at, say, influenza scaling trees. But people are trying to do that nonetheless.
Okay.
Yeah, great question, thanks.
Yep. Do we have any further questions? Feel free to unmute if you want to comment. And so if not,
I can ask one. Yeah. So Nigel, what do you think the ideas about universal biology, how they would help us, let's say, identifying life on Titan? Because you raised the issue.
Sure. So I can give you a concrete example of that. So I gave you one of the examples, which was this functional redundancy of metabolic systems. So let's suppose I want to ask the following question, which actually I do. And that is, one of the discoveries that Cassini made was that it discovered that there are global oceans of liquid water under the outer solar system bodies, in particular Europa, Enceladus, I think Pluto. The ones on Enceladus are the ones that are particularly interesting. Now, the way Cassini discovered that was when you look at the wobbling of the moon as it goes around Saturn, you can see that it wobbles in a slightly strange way, rather like when you take an egg that is hard-boiled and rotate it, have it spinning and then stop it, compared with one that is not boiled at all, which will then be able to rotate in a different way. When you stop it, it'll carry on rotating because of the motion of the liquid in between. And so by carefully studying the rotations of Enceladus, people were able, so I don't have the exact paper quote for you, were able to really infer that there was a global ocean of liquid water underneath the ice cap, and that is presumably melted by tectonic activity due to tidal forces from Saturn. So that's great. So you've got global oceans of liquid water, but the other thing you have is you have cracks in the ice. And so some of that water is squirting out into space. And so Cassini also went through that fountain and sampled the water chemistry. And so we know from the sampling of the water chemistry that it has a composition very similar to what you see in alkaline hydrothermal vents here on Earth, locations which are thought by some to be the possible origin of life on Earth. So now one might want to ask, well, what kind of life could exist in this kind of ecosystem on a planet like or a moon like Enceladus or Europa? So what you might say is, well, let me try to simulate the geochemistry and then get examples of those living organisms from Enceladus, put them into the geochemistry and see what kind of stuff comes out, see what kind of water chemistry they create, and then see if that's what we see from the Cassini mission. However, of course, we can't do that because we can't get those organisms to put into our laboratory apparatus. But we can create simulations of that on Earth by using terrestrial organisms that can live in those environments. And if you have reason to believe that there is functional redundancy and universality in the metabolic output, then the terrestrial organisms that can live in that same environment would have the same output as the Enceladus organisms that can also exist in that system. And so one can actually make meaningful predictions by using experimental systems as exemplars of what you might see in this astrobiology context. And so that's just a proposal for how one could exploit this understanding of universality to answer interesting questions in astrobiology. So does that answer your question?
Yeah, thank you very much. Fascinating.
Yeah. So there's one last question. So maybe we take it as the last question from Andreas Major. When thinking about biological systems and their response to perturbations, what measurable quantities can be used on the larger scales?
So let's suppose we look at antibiotic resistance. Let's take a very concrete example. So you invent a new antibiotic today. So congratulations. And you apply that, let's say to treat tuberculosis or gonorrhea or whatever it might be. So great, you spent $2 billion creating a new antibiotic. It works. 5 years down the road, you start to measure that the efficacy of that antibiotic has started to fall. And by the time, 5 to 10 years later from that, you find that the cases that present in the clinical setting, excuse me, the cases that present in the clinical setting are not cured by that antibiotic. So that's how you've measured how the system has responded to your intervention. You've applied a selection pressure in the system, and all you've done is accelerate the evolution of a superbug, or MSRA type of a bacteria. So that would be one way that you can track this process. So you may want to think of different ways to treat antibiotic infections. And actually, I proposed ways to do that by looking at the disease as an ecosystem and formulating ways where you can intervene in the ecosystem which don't involve applying a selection pressure. And actually, this is unpublished work that I never got around to writing up, but the work is in one of my students' theses. But one of the things that you can do, for example, is look at the ways that pathogens coordinate their release of toxins during a bacterial infection. And one can intervene with the quorum sensing communication channel. Now that's not a new idea. Many people have thought of the idea of stopping pathogens communicating by interfering with quorum sensing. But actually the way to intervene without creating a selection pressure is not to reduce the quorum sensing, but to actually increase it. In other words, to add autoinducers to the system. And what the autoinducers do is they make the pathogens think that they have a higher population than they actually do. And so then they release their toxins, which then causes them, there's a metabolic cost when they do that. And then the beneficial bacteria in your gut will then be able to kill the pathogens and wipe them out. So you can think of interventions, evolutionary interventions, that will be, pretty much immune, not completely immune, but pretty much immune to evolvability. So that's a concrete example. And the reason we didn't publish the work was we then wanted to do experiments for this. So we wrote proposals to do that. We had very specific experiments to do in vitro and even in vivo. But it was hard for us to get evolutionary proposals funded in the US National Institute of Health. So then we went on to other things and never got back to that. But there's an idea there which is very, I think, intriguing and is worth exploring.
All right. Well, thank you very much again for your time and for the really thought-provoking talk again. And yes, so thank you. Okay, well, thank you. I'm very happy to have met some of you. I'm sorry that I have to dash off. I hope this talk gave some of you, particularly any students, postdocs who are here who want to think big, as it were, or think beyond what they're doing on their day-to-day basis. Hopefully that will stimulate some discussion and activity, and I hope you found it interesting. Yeah, I'm sure it has. Thank you. Bye-bye. Okay. Bye-bye, everyone.
Bye.